Hello, I'm Rollins, and welcome to Pharma Focus Europe. This is your gateway to the latest innovations in pharmaceutical industry, biopharma breakthroughs, clinical trials, and advanced manufacturing. Whether you're working in research and development, regulatory affairs, biotech, or anywhere in between, stay connected to pharma innovation anytime, anywhere with Pharma Focus Europe. Want more? Subscribe now to www.pharmafocuseurope.com/subscribe and get the latest updates delivered straight to you.
Hello listeners, today we're diving into a powerful and evolving topic, from data to decision leveraging protein-protein interactions for safer polypharmacy. As polypharmacy becomes more common, so does the risk of adverse drug interactions. Today, we explore how mapping protein interactions is revolutionizing drug safety, turning complex data into smarter, safer prescribing decisions. Today, we're joined by a dynamic team from Indian Institute of Information Technology, Allahabad. First, we have Dr. Samantha, Assistant Professor, whose work merges data science and biotechnology. Next, we have Ananya, an expert in applied sciences, and Dr. Sarfraz Anwar, specializing in protein-protein interactions. Together, they'll share insights on leveraging protein interactions for safer polypharmacy, a game-changer in drug development and therapy. Let's dive in. Together, this trio has proposed a significant shift in how we model and predict side effects in multi-drug therapy, one that brings us closer to safer, smarter prescribing. Now, let's dive into the findings and explore how protein-protein interactions could reshape the future of personalized medicine. From data to decision leveraging protein interactions for safer polypharmacy. Polypharmacy, the use of multiple drugs simultaneously, poses significant risks of adverse interactions. To mitigate these risks, improved AI-driven frameworks are essential for predicting side effects. While research has utilized machine learning models like artificial neural networks and XGBoost to assess drug-drug and drug-protein interactions, protein-protein interactions remain unincorporated in datasets. Addressing this gap could enhance prediction accuracy, highlighting the need for further development of AI and ML models in polypharmacy side effect prediction. In today's rapidly advancing field of modern medicine, the practice of prescribing multiple medications at once, referred to as polypharmacy, has become increasingly common, especially for individuals undergoing chronic or complex health conditions. While intake of more than one drug is essential for disease management, it also introduces a significant risk, the possibility of harmful drug interactions leading to serious or even life-threatening side effects. As a result, predicting these interactions has become a crucial focus in healthcare, driving the development of advanced AI and machine learning models designed to anticipate these dangers. Traditionally, these models have been centered on understanding either drug-drug or drug-protein interactions, utilizing extensive data sets to identify potential risks. However, as our understanding of cellular biology deepens, it's evident that these models may be missing a critical component: protein-protein interactions. Proteins don't function in isolation, they interact with other proteins, forming complex networks that are essential for biological processes. Disruptions in these networks, whether due to disease or drug effects, can lead to a cascade of unintended consequences that traditional models might overlook. By incorporating PPI data into our current frameworks, we're on the cusp of a significant breakthrough in predicting polypharmacy side effects, which could result in safer, more personalized treatments, bringing us closer to the goal of truly individualized medicine. Integrating protein-protein interactions into AI/ML models. Incorporating protein-protein interactions into AI and machine learning models offers a promising way to enhance the accuracy of predicting side effects from polypharmacy. By taking into account the intricate network of protein interactions within cells, these models could more accurately forecast how a combination of drugs might disrupt normal cellular processes. For instance, a drug that impacts a crucial protein involved in multiple PPIs could trigger wide-ranging effects that current models fail to predict. To achieve this integration, researchers can access existing databases and tools that document PPIs. Resources like the String database and BioGrid offer extensive datasets on known and predicted PPIs. By merging this data with existing drug-protein interaction datasets, AI/ML models could produce more comprehensive predictions, ultimately reducing the risk of adverse drug interactions. Developing models that effectively integrate protein-protein interactions into AI/ML frameworks for predicting polypharmacy side effects involves a multifaceted approach that combines advanced computational methods with extensive biological data. Data integration and pre-processing. The success of these models depends on effectively merging various types of data, including drug-drug interactions, drug-protein interactions, and PPI networks. One of the main challenges is dealing with the diversity of data sources. While DDIs and DPIs are typically available in well-organized databases like DrugBank and PDDID, protein-protein interactions are often gathered from platforms such as String, BioGrid, and Intact. To build a comprehensive dataset, this data needs to be normalized and standardized. Techniques like ontology mapping and using standardized identifiers, such as UniProt IDs for proteins, are essential to create a unified dataset that ensures compatibility and accuracy. Graph-based model architecture. One effective approach to developing these models involves using graph-based algorithms, where proteins and drugs are treated as nodes within a network, with edges representing their interactions. Graph neural networks and their variants, like graph convolutional networks or graph attention networks, are particularly well suited for this purpose. These models are capable of capturing the complex relationships and hierarchical structures found in biological systems. For instance, GNNs can spread information across the graph, enabling the model to learn from both direct and indirect interactions. This allows the prediction of how a drug might disrupt a PPI network, potentially leading to adverse effects. Feature engineering and representation learning. The success of these models largely depends on how accurately the biological entities, like drugs and proteins, are represented within the model. This is where feature engineering becomes crucial. For PPIs, features might include factors such as interaction strength, binding affinity, and post-translational modifications. Representation learning techniques, such as embeddings, can be used to convert these features into dense vector representations that capture the underlying biological relationships. For example, protein embeddings generated using techniques like Word2Vec applied to protein sequences or autoencoders trained on PPI networks can offer rich, context-aware features that significantly improve model predictions. Transfer learning and multitask learning. To boost the model's effectiveness, techniques like transfer learning and multitask learning can be applied. Transfer learning enables the model to use knowledge from related tasks, such as predicting DPIs, to enhance its predictions on PPIs. This approach is particularly valuable when PPI data is limited or noisy. Multitask learning, on the other hand, involves training the model on several related tasks at once, like predicting side effects based on both DPIs and PPIs, so the model can share the learned representations across tasks. This strategy can lead to better generalization and increased robustness in predictions. Model validation and interpretability. A key part of developing these models is making sure they are both valid and interpretable. Cross-validation techniques, such as k-fold validation, are crucial for assessing how well the model performs. Additionally, interpretability methods like Shapley additive explanations or integrated gradients can help pinpoint which features or interactions are most influential in the model's predictions. This not only helps in verifying the accuracy of the model's predictions, but also offers key understanding into the biological mechanisms behind polypharmacy side effects. Predicting polypharmacy side effects is a complex yet essential challenge in modern medicine. Although AI and ML models have made considerable progress by factoring in drug-drug and drug-protein interactions, the omission of PPIs remains a significant gap. Integrating PPI data could enhance the accuracy of these models, minimizing the risk of harmful drug interactions and opening the door to more personalized treatment strategies. As research in this field advances, incorporating PPIs into prediction models is likely to become standard practice, bringing us closer to the ultimate goal of truly personalized medicine. Thank you for listening to our special feature titled, From Data to Decision, Leveraging Protein-Protein Interactions for Safer Polypharmacy, authored by Dr. Samantha Ananya and Sarfraz Anwar from Indian Institute of Information Technology at Allahabad. We hope this insightful exploration into predictive pharmacology and AI-driven healthcare has given you a deeper understanding of the future of personalized medicine. Stay curious, stay informed, and join us next time for more cutting-edge stories from the world of science and innovation. Stay tuned for more episodes, where we'll continue bringing you perspectives from leading voices and top executives across the pharmaceutical industry. Thanks again for listening to Pharma Focus Europe.